Hi everyone, welcome to case study number 1. I hope you had a chance to read through this slide and think about the scenario, as well as find the example test data set that was provided for case study 1, which in this case just consisted of a single sequence. And further, I hope that you were able to run that sequence through SeqScreen yourself or download the pre-computed outputs to interpret. And as you were looking at the outputs, I hope that you had a chance to think through what was going on your own. And now we are going to go through and hear what I thought about the outputs and my thought process as I was trying to figure out what was going on with this sequence. I also wanted to mention that this is a real sequence that was provided to me from a bioengineering and biotechnology company. It was something that one of their customers was ordering, using, and they wanted to know what our software could do to analyze the biosecurity concerns that might be present within this particular sequence. It was a challenging sequence, so other bioinformatics software packages were struggling with it, and it was an interesting use case that I thought would be worth mentioning. I did keep the company's name out of this case study, and I also was pretty vague and made up the scenario and experiment surrounding why this sequence existed, but that was to protect the anonymity of the company. They did give me permission to post this publicly and use it in the course. So thank you to them for submitting this and sharing it, and let's jump in. So I am going to open up the spreadsheet where we have pasted the outputs from the SeqScreen final report with the pathgo.tsv extension. So this is similar to what we looked at in the known sequence exercise, but now this is the output for case study 1. And I have a number of different tabs in the spreadsheet. If you downloaded it, looked at it on your own. Some of the tabs correspond to the different modes of SeqScreen that I ran in this case. Others correspond to intermediate files that came with the different modes that I thought might be helpful to look at. And we'll talk through all of that as we go. I just wanted to give you an overview of what's in this spreadsheet and that there are multiple tabs for a reason. I wanted to keep things separate and look at things one by one because that is really how the output files are generated. They're all separate output files. Okay, so in fast mode, when we look at these outputs, we see that there was only one assignment made by Diamond, and it is a particular protein from yeast. And that protein had a pretty good E-value. It did not have any pathgo terms assigned, and there were no fun socks. So no obvious pathogenic functions here. Although if you remember from my previous discussion about pathgo, if there is a dash there, that means that it wasn't annotated by our biocuration team. And I can tell you that the reason that this one wasn't annotated is because they really focused their annotations on proteins that represent sequences of concern. And this is not a sequence of concern. And that's not to say that everything they have not annotated is not of concern and everything they have annotated encompasses all sequences of concern. But in this case, that's why they didn't annotate it. And so nothing concerning here in terms of the protein itself. The only thing that jumped out at me was this, that under the multi-taxide confidence field, we see that the value for the confidence is 0.578. And that is relatively low on a scale of 0 to 1. That's about right in the middle, which is not a very high confidence. So that's something that I found strange and is worth looking into further. So the next thing that I did is that I looked at the HTML report output for fast mode, and that is what this is that I'm showing you now on the screen. If you go to the HTML report and you click on the query sequence, you should see the alignment visualization for this particular sequence against the best reference that it found. And if you are familiar with alignments, you'll notice immediately that there's a very poor alignment in the middle of this sequence. So that's this area right here. If you're not familiar with alignments, a good alignment will show that the same amino acid abbreviation is present between the query and the subject, and that is denoted by the same amino acid letter showing up in the middle here. So this is all a perfect alignment, and this is a perfect alignment at the beginning and the end of the sequence, but the middle is not. We see one shared T here, and a few pluses, which means that the amino acids shared some commonalities, but they were not equivalent. So not a great alignment in the middle of the sequence to yeast. Something else is going on here in the middle that is not yeast. So to further analyze this, I ran sensitive mode. And before I move on to the sensitive mode results, I should mention that I also included the fast mode diamond outputs here. So these are intermediate files that you can find in your seq screen output folder within the taxonomic identification subdirectory. And then within that subdirectory, you go to the BlastX subdirectory, and that's where you'll find these outputs. This is the B tab output, BTAB, and it essentially has all of these different fields of output that you would expect for a BlastX kind of result, which is what seq screen reports for Diamond in this case. And I added the headers to this particular file for ease of interpretation during the course. And hopefully that helps everything make a little bit more sense. The 0.578 number that we were talking about previously that I said looked strange for the confidence of the multi-taxity, so the confidence for this being yeast was really weak. That is reflected here. Other things that you might pay attention to include the E value, and the bit score. These are all significance indicators. So that's our diamond output. Fast mode also runs centrifuge, but in this case, centrifuge did not come to any good conclusions about the sequence. It was unclassified. And I think it's worth noting that the centrifuge database that we use with seq screen is only a microbial database. So if any of the sequences come from eukaryotes or other non-microbial organisms, that will not appear in the centrifuge database, and it will not be able to be detected with centrifuge in the way that we're using it here. Our diamond and blast databases that are used with seq screen, so the big protein database and the MP database, those are all moving beyond just microbes. We have all kinds of things there. So they should be able to be more broadly detecting different toxins, whereas centrifuge will just be focused on microbes. Okay, moving to the sensitive results, I jumped right to the detail here. So we're looking at BlastX outputs for sensitive mode. These are very similar to the diamond outputs, but one of the first things you might notice here when comparing BlastX to diamond is that diamond only have one row, whereas BlastX has a lot of rows. That's because BlastX is, as the name would suggest, more sensitive in sensitive mode, and it's going to return more results. Diamond runs very rapidly and efficiently. BlastX is going to be more detailed in the number of outputs that it can provide in the way that we run it in seq screen. And we see that reflected here. So one thing that jumped out to me as I looked at this is that the first row in the BlastX output is identical to the first row in diamond. So this is the same protein that's being returned on top. The next protein though looks not like a terrible hit either. I mean, the percentage of identical matches is fairly high. The expected value or E value looks, pretty comparable between these two. The bit scores look pretty comparable. So as far as strength of the hit, these first two rows look like they're about the same strength. Some differences, but both would be worth paying attention to when we're trying to figure out what's going on here as I glance through this. And when I look at the actual name of what these proteins and information about the sequences might be, we see that the first one is the yeast protein that we've talked about before. The one below it is actually a snake toxin. So this here is a protein associated with a snake toxin. So that's interesting from a biosecurity standpoint. It's worth mentioning that the list of regulatory organisms and toxins does not include eukaryote toxins like snake toxins. But from a biosecurity standpoint, it's something worth paying attention to. If you weren't expecting this customer to be working with something like a snake toxin, that's going to raise a red flag, I think, in my mind, of just asking questions and figuring out what's going on here and making sure that it's safe and appropriate for use. In the case of the company that provided the sequence as they're analyzing this for one of their customers. So there it is. Another thing that can be analyzed here, just to clarify where these particular matches appear in the alignment, is this start of alignment inquiry and end of alignment inquiry field. So for the top row, which is the yeast, we see that it starts at position 3 and ends at position 137. The reason for that, which is that, by the way, is like the full length of the sequence, more or less. The reason for that is that when we think about alignments, it's returning the beginning of the alignment and the end, and then it's not necessarily worrying about the middle part not being aligned within the metric that we're describing here, where we're talking about start and end. Just where did it start, where did it end? For yeast, it started here and it ended here. However, for the snake toxin protein, it starts at position 48 and ends at position 116. So that's actually closer to the middle of the sequence. And so there you have it. We have yeast flanking a snake toxin protein. I went ahead and analyzed it with the other mode in seq screen just for fun, see what would happen. I also included the blast end results here from sensitive mode, which are primarily yeast, but you can see that the yeast is showing up at the beginning and the end of the sequence here in the blast end results. For ONT mode, it was interesting to me that in ONT mode, it really keyed in on this middle snake toxin sequence. And I think the reason for that is that ONT mode is made to look at long sequences and try to infer the open reading frames within them. And since this middle sequence was its own open reading frame of a particular protein, it focused on that in its reporting here in the ONT mode seq screen results. And we can look at how the pathogenicity labels look for this particular snake toxin. We see that disable organ is the fun sock that's assigned. So that's the most severe, most damaging fun sock possible within seq screen. And then we also see a pathogen here at the end, and that is suppresses coagulation in another organism. So the fun socks were assigned by machine learning. So the machine learning algorithm assigned disable organ, whereas a subject matter expert assigned the pathoterm suppress coagulation in another organism. And that subject matter expert biocurator also assigned a PubMed ID, which is a reference to a scientific publication where they provide functional evidence for why that particular pathogenic function was assigned to that protein. And if we jump to that particular publication, we see this. And so if you are using seq screen and you see a pathoterm like this and you want to learn more about it, you can go to this publication and learn more about the experimental and functional evidence for the pathogenic activity of that protein. So here's this. That's helpful, I think. And then the final tab on the spreadsheet is giving you more detail about the ONT diamond outputs, which are just the outputs that correspond to the snake toxin protein that was identified in the middle of the sequence. Hopefully that was helpful. I think that overall SeqScreen provided the information that was needed to figure out that this is what we refer to in the bioinformatics community as a chimeric sequence. So it's a sequence with more than one organism present within it. So there's more than one protein from different organisms in the sequence. And that is challenging because you're not getting, in this case, a really long sequence that's contiguous from one organism. You're getting small pieces that are mixed up, and that often confuses our algorithms and our reporting. So it is a tricky case, but I think SeqScreen did a pretty good job in explaining what was going on. It did take some looking. We had to look through the results and think about it. But I think overall, I was pretty happy with its performance in this case. So I hope that was helpful and you enjoyed case study 1. All right, thank you so much for your time.